It is my very great pleasure to introduce our plenary speaker today, Professor Rob Nicholls. Rob was the catalyst for this particular meeting and this particular timing, and I think he'll agree that it was an awesome chemical reaction and that we have catalysed the presence of a huge number of people from a very diverse set of research and lived experience backgrounds, which is awesome. So Rob is a professor at the University of Pittsburgh in the US. He is an absolute legend in the field of Prader-Willi research. It was a critical role in discovering imprinting and was the first to characterise the UPD genetic subtype. So it's my very great pleasure to introduce Rob Nicholls. He's speaking on an odyssey in PWS research from imprinting genes, animal and cell models, and genetic therapies. Welcome, Rob.
Thanks very much. It's a pleasure to be here, and I think some may not know, but I'm Australian. I grew up here in Victoria, moved around quite a lot, different towns. I was born in Ballarat, lived in Denali, Yarrawonga, Neerim South, Broadford, and finished high school in Inverloch, which is where I consider my hometown. So there's a pocket of Australian Prader-Willi research in Pittsburgh, and I'll tell you about some of the work we've been doing today. Let's see if this will, maybe this one. Okay. So quick summary, I'll talk just a little about human genetics. I actually spent the first 10 to 15 years of my career working on Prader-Willi syndrome doing human genetics. That's how I trained. But then we transitioned into animal models, studying mouse model, Prader-Willi, subsequently a pig. And we use animal models to try to get at tissues and physiological functions that we just can't do experimentation with humans. And when you're talking about the brain, other organs, and a process, we'll talk about genomic imprinting, which occurs in germ cells and early pre-implantation development, then that's why we need other model systems. We've also made use of cell models. In cells, we can manipulate genetic functions much simpler, and I'm going to talk about predominantly studies on insulin-secreting beta cells. We've also made a growth hormone secreting somatotrope Prader-Willi model. And the goal of the research is to better understand the pathophysiology of Prader-Willi syndrome, and then to hope to use that knowledge to translate to therapeutic approaches, whether that's new small molecules or drugs or genetic-based therapies. And my lab's taking one direction. We're going to hear today, I think, about other directions too, to reactivate the silent genes. Okay, so my early work, I started work after doing my undergraduate degree here at the University of Melbourne. I went overseas and went to England and did my PhD. That was in the molecular basis of alpha thalassemia as a blood disorder. But I was always interested in brain disorders and genetic conditions. And so then I moved over to the US and did a postdoctoral research with the late Sam Watt at Harvard University Children's Hospital, Boston. And that's where I started work on Prader-Willi syndrome. It was, at the time, all we knew was that chromosome 15 was involved. There was a deletion in maybe half the patients that David Ledbetter had observed under the microscope. But there was no DNA studies, no genetic studies. And that's what I set about initially to develop, was genetic markers. And I'm not sure if my pointer's working. But on the left here, we see a DNA analysis of a child with Prader-Willi syndrome, the mom and the dad. And this is a DNA analysis that we can separate the different chromosomes, the two chromosomes from the parents. And you see that mom has two different bands, and dad has double intensity of this upper band. The child with Prader-Willi only has the band shared with mom, so the band from dad is missing. And this is what we found in all the deletion patients, that the deletions were derived on the paternal allele. And there's another genetic disorder, many of you have heard of, Angelman syndrome. They have the same deletion, turns out, but on the maternal allele. But we had a large number of families, still about half, that didn't have a deletion. And there were also reports in the literature that there were maternal deletions in Prader-Willi. Well, they turned out to be incorrect reports. They actually were Angelman, but they hadn't been recognized because of the clinical overlap in that early newborn period. But in studying one of these non-deletion cases, you see here, this is a more complicated DNA analysis in which we have 3 bands. We have this band, this one, and this one. Dad has 2 copies of this one. The mom has the 2 different bands, and the child with Prader-Willi has the same 2 as mother. And so this is what we call uniparental disomy, 2 maternal copies. And on the right, we see the chromosome 15, and on the left is the deletion subtype with a deletion on the paternal-derived allele in about 2/3 to 3/4 of patients. And then on the far right, in UPD, or uniparental disomy, there are 2 copies of the chromosome 15 from the mother. And so in both cases, it's the loss of paternal gene expression that leads to Prader-Willi syndrome. And this just shows, one thing that we've learned about Prader-Willi from the start is that whatever you discover, it's more complex than that. But at the same token, Prader-Willi syndrome has also been incredibly important for understanding human genetics. We've learned a lot of human genetic principles from study of Prader-Willi syndrome. And so this slide shows not just the deletion group and the uniparental disomy group, but there's a third class called an imprinting defect, in which there's both a paternally derived and a maternally derived chromosome, but the paternally derived chromosome has a defect that leads to a maternal imprint. As I say, Prader-Willi is always more complicated, so this class actually splits into 2. There's a familial group, and they have very small mutations, small deletions that define a genetic element that controls the genomic imprinting process. And there's a sporadic group which have no mutation at all. And it turns out that that sporadic group arises from either just a chance occurrence or an environmental effect early in the pre-implantation embryo before the 8-cell stage. It's at the 8-cell stage that the imprint is finally fully established. And then there's a very small subset of a handful of patients that have very small deletions of a set of what we call SNO RNAs, or small nuclear RNAs, and I'll show that in a slide in a moment. There's actually about 12 genes that are involved in Prader-Willi syndrome, so it's much more complex than a lot of genetic disorders. We heard earlier about cystic fibrosis, a single gene, Angelman syndrome, single gene. Single gene disorders are still complex to try to develop therapies, and for many, they're still undergoing. Well, here we have these 12. There's actually 10 highly conserved, so I consider 10 genes that are particularly important. So I talked about imprinting already. What is that? It's what we call an epigenetic phenomenon, which is the way that genes are regulated and their function dependent on the sex of the parent of origin. So imprinting is a reversible process. It goes through a life cycle where adults, where individuals have both a maternal and paternal imprint, but then in the germ cells, they have to reset the imprint, so the imprint's erased, and the reset specific for, in a male during spermatogenesis, the paternal imprint set, and in a female during oogenesis, the maternal imprint set. And then after fertilization, they come back together and you have the two chromosomes. And Prader-Willi syndrome was the first recognized human disorder affected by genomic imprinting. There are multiple others now, and also a number of childhood cancers that involve imprinted genes. And epigenomics or epigenetics is the regulatory information that controls the gene expression. And we will hear about that also later today. So this is a gene map of the Prader-Willi region. It's complicated. All the genes, all those little symbols in blue are paternally expressed genes. They're only expressed after inheritance from the father. The circles are protein coding genes, and the very small, thin ovals are RNA genes. Many of them, there's a cluster of 5 snow RNAs or small nuclear RNAs. Some of these, a single copy in the middle, 107, 64, single copy. SNORD 116, there's about 25 copies in humans. SNORD 115, there's about 50 copies in humans, so these are amplified. And the key protein coding genes, there's SNRP, Nectin, MAGEL2, McLaurin3. The imprinting defect, familial cases that have small microdeletions have microdeletions right here in the promoter and enhancer of this bicistronic SNRP gene, and that sets the imprint for that entire 2 megabase region. And then the promoters of this bicistronic gene also drive long transcripts of a couple megabases in neurons that the small RNAs are then processed from. So it's a very complicated genetic region. The Angelman syndrome gene, UBE3A, is on the right there. It's only expressed out from the maternal allele in certain types of neurons. And then the patients with deletions have deletions from either this orange or this orange zigzag line through to breakpoint 3. And the genes in white are non-imprinted genes, so they may be associated with other recessive disorders. For example, the OCA2 gene is involved in oculocutaneous albinism and is associated with hypopigmentation, so reduced pigmentation in the skin, the hair, and the eyes in patients with deletions. Okay. And this is just to highlight the instability of this chromosome domain. These breakpoint hotspots not only occur with deletions in Prader-Willi and Angelman syndrome of 2 types, but there are larger duplications and triplications or even isodecentric chromosomes that have the same region repeated. And these are a common event in autism, one of the 2 most common genetic events. And then there's other copy number variation of these regions that don't involve the Prader-Willi gene. So it's a very complicated genetic region that leads to the instability that gives rise to the deletions. We're going to transition and talk about some animal models now. And we use animal models to understand what causes the disease when we can't access tissues in human. It also, animal models may be important as preclinical models to look at new therapeutic approaches, particularly if it's a non-standard drug that's never been tested in humans. We're going to talk first about the rodent, the mouse or the rat, and you'll see that the PWS genetic map looks pretty similar. There's one extra imprinted gene in rodents that humans don't have. It's expressed at low levels. It's probably not that important phenotypically. And rodents are also missing SNORD 109, which has 2 copies in humans. Otherwise, the genetic map is pretty much the same. And so in my lab, we studied a mouse model that has a large deletion shown at the top of all the same genes as in Prader-Willi syndrome. And then I'll show some studies in a rat beta cell model in which we made a deletion using genome editing of just the paternally expressed genes. So equivalent deletion to that. So this is the Prader-Willi mouse model in the middle. The little Prader-Willi mouse is in dark fur, and the wild-type littermate is white fur. They're 5 days of age. On the left here is the growth chart, and just before birth, the Prader-Willi pups are slightly smaller, significantly so. Postnatal day 1, they're smaller, and by day 5, they're half the size. You can see that the Prader-Willi pups are still growing, and they do have milk in their bellies, so they're absorbing some nutrients because they wouldn't grow otherwise beyond about P1. But if you look at the right, we see the survival, the Prader-Willi curve is in blue. And you can see by day 4 or 5, beyond that, we lose all the pups. So they have failure to thrive. There's much less muscle mass. They have more difficulty feeding, and they don't survive. This is true of the deletion mice or imprinting defect mice too. So the reason they don't survive, we wanted to figure this out and did a metabolic and endocrine workup. And what was really interesting was the glucose levels, the blood glucose. So just before birth, blood glucose was normal. At postnatal day 1, it was normal. But by day 2, the Prader-Willi mice became hypoglycemic. That progressed. And by day 5, they had almost no blood sugar. Well, your brain can't function without blood sugar. And so it was clear that they were stopping breathing, seizing. That was why they weren't surviving. We looked at ghrelin. Many of you heard of ghrelin, a hormone released from the gut that is involved in feeding. It was thought that it may play a key role in Prader-Willi when it was recognized that ghrelin levels were really, really high in Prader-Willi. What we found in this mouse model was that ghrelin levels were normal when blood sugar was normal. It was only when they became hypoglycemic that ghrelin levels went through the roof. So I think of ghrelin as a physiological marker of hypoglycemia, and the sugar level, blood sugar level, is your energy level. It's actually been shown in mouse models that hypoglycemia induces hyperphagia. And so there could be a link still that we don't understand between hypoglycemia and hyperphagia. It's important to point out that when we found hypoglycemia in the mouse, it wasn't known in Prader-Willi. And then Jen Miller, a pediatric endocrinologist at the University of Florida, and her students went back and looked retrospectively at charts and found that infants with Prader-Willi were in fact having quite severe episodes of hypoglycemia. And that's still under-recognized clinically. A lot of my clinical colleagues who work with young adults with Prader-Willi have said, oh yeah, we find episodes of hypoglycemia at times of stress. And so I think it's still an area of Prader-Willi research clinically that needs to be further investigated, because blood sugar is really critical for during neonatal and postnatal brain growth and function. So, okay. So we also found that blood circulating levels of the hormones insulin and glucagon were low in Prader-Willi mice compared to wild-type mice. Insulin and glucagon co-regulate blood glucose levels. Insulin allows for the uptake of glucose in muscle and other tissues, and glucagon's counter-regulating that response. So those levels were low, so we said, well, let's look at the pancreas, and these studies are all done at postnatal day 1 before they become hypoglycemic. And this is an immunostaining of a section of the pancreas. This is a wild-type pancreas in the mouse. The beta cells are in green, and you have this nice spherical islet surrounded by glucagon expressing the pink cells, and it looks really pretty. This was the Prader-Willi eye. It looked like you took a wet towel and wrung it up, and so the alpha cells are intermingled in with the beta cells. Was just completely disrupted, and this actually we found in the fetus and postnatally. There was also a 50% decrease in alpha and beta cell mass, and an increase in apoptosis, cell death, in alpha and beta cells that contributed to that failure. So then we took the islets out of the mice and cultured them in vitro and did an insulin secretion assay. And at both low glucose, which is the basal level of insulin secretion, and with glucose-stimulated insulin secretion, the Prader-Willi islets were greatly defective compared to wild type. The Prader-Willi islets are not defective in glucose-induced secretion because they increase at a similar rate to the increase for wild type, but the basal mechanism is defective. And on the right, we did another experiment where we bred on a fluorescent protein transgene marker of insulin secretion. And this was a unique fluorescent protein that changes color from green when first synthesized to red over 24 hours. And so what we see here on the left is the control mice in which the beta cells are mostly green with a little bit of yellow, showing that it's newly synthesized insulin, and insulin secretion occurs right after from newly synthesized insulin. Whereas on the right, you see that the PWS beta cells are mostly red. So the insulin is aging and not being secreted. It's sitting there in cells. So there's an insulin secretion defect. So we then had a bit of trouble with NIH study sections trying to get the mouse model funded. They didn't understand the imprinting, the complexity of all the genes. So we ended up turning, once genome editing was developed, to developing a cell model, and we just published this a couple months ago in PLOS Genetics. And I'm not going to go through the details here, but basically we deleted the Prader-Willi genes and then selected for cell lines that were missing the Prader-Willi gene expression. We generated 3 Prader-Willi cell lines, independent cell lines, and 3 independent control cell lines. We used this cell model to do the insulin secretion assay at both low and high glucose, and you see that Prader-Willi cells are deficient in insulin secretion, but they react to glucose. This looks exactly the same as from the mouse. So this insulin secretion defect is a cell autonomous defect. It's not dependent on the physiology of the animal. It's purely a cell-derived mechanism because of the Prader-Willi defect. So we then looked at 2 types of assays, looking at every protein expressed in the beta cell and every RNA expressed. And this is the proteomics assay. And we analyzed 3 types of proteins. We looked at the insoluble large protein fraction, the soluble small protein fraction of cells, and then what was secreted. And we got the same answers in all cases. So that in red, the hormone secreted from the beta cell, insulin, another one called amylin or AAPP, and they're greatly reduced. This is the large protein fraction, so these are residual peptides. This is the small protein fraction. You see here, this is the amount of the protein expression, so there's a lot of the small proteins, but this 0 line is the average. So anything down here is reduced. And then the same thing for the secreted. All the secreted peptides, insulin and amylin, were decreased in Prader-Willi. But the big surprise and the new finding was that when we looked at the large protein fraction, all these proteins in green, particularly these 2, which are highly expressed, but all these other proteins in green are a family of 10 to 12 endoplasmic reticulin chaperones. And these are proteins that are critical for folding proteins that are being secreted or sent to the cell surface. And insulin is one of the hardest proteins there is to fold because of the way the hormone is constructed. And so this actually helps explain why we're seeing insulin aging and not being secreted, because the proteins that are meant to be there to help fold it and traffic it are reduced in size. And we confirmed all this. I won't go through the details, but we used Western blotting using insulin at top, M-cherry, which was a marker we had of insulin secreting cells, and then the ER chaperones. And they all showed that the Prader-Willi cells were deficient in these proteins. And we also looked at every RNA. David will like this slide. I won't go through all the details, but looking at RNA sequencing, all these genes in purple that on this volcano blot are completely downregulated. These are the Prader-Willi genes. You expect them to be gone. They are. The genes in green are these ER chaperones, which are reduced, and the ones in red are the hormones. We found some proteins that were upregulated. We haven't yet studied these in great detail. And then we confirmed everything with droplet digital RT-PCR, and we don't need to go through those details. So 2 of the major ER chaperones are called HSPA5 and 90B1. And when those have actually been knocked out in the mouse, and in each individual case, they lead to decreased insulin secretion. But then there's dosage compensation so that other ER chaperones are increased to help counteract that. That can't happen in the Prader-Willi beta cells because all the ER chaperones are down. You can't upregulate any of the others. And so we hypothesize that a chronic shortage of ER chaperones in PWS beta cells leads to deficiency in the folding or delay in transit through the endoplasmic reticulum to the cell surface, and that's why insulin is defective. And so one question is this also the basis of other hormone defects in Prader-Willi? Does this affect growth hormone secretion? Does it affect other hormones like oxytocin, which is lower in Prader-Willi, and a number of other gut and brain and neural peptides? We don't know the answer to that yet. And so I think I've said this. Many of the features of Prader-Willi could be explained by deficits in protein secretion. And what this shows is that the Prader-Willi genes that are paternally expressed normally function in late fetal and postnatal neurons and endocrine cells. And therefore, this is a stage that's critical for transition of the fetus to independent life. And so it appears that Prader-Willi genes are affecting that transition from the fetus to independent life and survival. Okay. And I won't go through the details here, just to show that the ones here in the middle, which are missing Prader-Willi genes, these are somatotrope Prader-Willi cell lines that are deficient in secretion of growth hormone. So we're now looking at RNA sequencing and other techniques to see what's deficient there. And so overall, there are a number of hormone systems affected in Prader-Willi. We know that a number of neuropeptides, we don't know exactly which ones that may play a role in hyperphagia, are deficient. Growth hormone is deficient from the pituitary. We know that Prader-Willi individuals have hypergonadotropic hypergonadism, so that system's deficient. We've shown that pancreatic hormones are deficient, oxytocin is deficient, ghrelin's overexpressed, which again may be a physiological mechanism. So the mouse models have been helpful, but the complete genetic Prader-Willi mouse models don't survive more than a couple of days, and so we can't model hyperphagia or behavior because the animals don't survive. Many workers have generated single gene knockouts for the individual Prader-Willi genes in the mouse. They only have mild phenotypes and only on an inbred background. So, they're important for showing what Prader-Willi genes may do and contribute to physiologically, but they're not showing the full picture. And another point is that mice and other rodents are lissencephalic. They have smooth brains. They're not going to be the best models for studying higher behavioral analysis. So we want a mammalian model with a gyrocephalic brain that more closely models the neural capabilities of higher primates. So this is critical for behavioral phenotypes. I just show this slide. So this is actually a rat brain. So it's smooth. This is the human brain. You see how folded this cerebellar is. Through the cross section, you see the same, the smooth brain and the other. If you look at a couple of other species, you'd say this brain is much more like a human brain size, gyral complexity than this brain. And what's interesting is that this is the non-human primate, the rhesus monkey, which many workers want to study. This is the pig. So this would suggest that pig may be better for neurobehavioral studies than even a non-human primate like the rhesus monkey. And so we set about, first had to build the genetic map, because when I started this work, the pig genome was just starting to be sequenced. Luckily, I was able to tie in that. They sequenced all the different backgrounds for me. You see that the organization of the Prader-Willi domain is essentially the same. The imprinted control region. Pigs don't have SNORD64. That's the one SNO RNA that's missing. And what we did was we made mutations in the imprinted control region to generate a Prader-Willi model. Just quickly, we're able to use a set of somatic cell hybrids that retain either the paternal or the maternal chromosome encoding the Prader-Willi genes. And this is the normal band. These are the genome edited band in different sets of guide RNA. So we're inducing the mutations at high frequency. But when we actually used a set of somatic cell hybrids that had the maternal allele, most of the guide RNA pairs didn't work. Only one pair worked on the maternal chromosome. So the other guide RNAs couldn't access the closed chromatin. And so we used this set to generate models because we wanted to try to generate deletions on the maternal allele. So we first injected, this was done in collaboration with a group in Missouri, and we first wanted to check if this is working in vivo. And so you inject the RNA components into the fertilized egg, and same technique as they used to make the COVID vaccines. And culture in vitro for a week, then we isolated the genomic DNA and did the analyses, and we see all these bands here, the breakpoints, so it's working in vivo. It's expensive to do the next step, and so, which is to repeat the process, but instead of isolating DNA after a week of culture, we implant those embryos into a female and carry litters to term. We obtained 2 litters. This female had a inversion on the maternal allele, and these 2 piglets represent 1.5 Prader-Willi pigs. This guy had hypertonia, has the full genetic Prader-Willi. This guy, turns out, he's a mosaic. 50% of his cells have the Prader-Willi deletion, and the other 50% are wild type in function. They actually have this inversion on the maternal allele. So we have 1.5 postnatal growth retardation. The fully affected pig had severe hypoglycemia. We were able to treat him with salt carb and keep him alive. The partial Prader-Willi had mild hypoglycemia. He did fine, and then they grew up. The Prader-Willi pigs on the right, you see his severe scoliosis, so this looks like it could be a good model to study scoliosis in Prader-Willi and to try to improve surgical or other techniques to overcome the scoliosis. Started off, these pigs were first generated in our collaborators in Missouri, and they kept them from the beginning on a very strict maintenance diet. And so the Prader-Willi pig is actually in red. He's a little smaller than his wild type, than his mosaic little mate. And so they grew, but then when we shipped them to Pittsburgh, my vet said, oh no, we can't control the diet, let's let them feed. Well, their weight just took off. And they freaked out and said, no, we've got to control the diet. And so then it settled right back down. So we think they have hyperphagia, but we have an N of 1, and the comparison's not even a control, so we're not certain. So for now, we've found that the Prader-Willi pigs, they have muscle hypertonia, failure to thrive, hypoglycemia. As an infant, the full Prader-Willi had a couple episodes. As an adult, we think he had a viral infection or something. Set off, we had to make him a little piggy milkshake with like Ensure and other things, and to get him to feed. The interest, there's scoliosis, and interestingly, the Prader-Willi pig had problems with anesthesia and temperature regulation. He would not be able to control his temperature and would go into hypoglycemia when we're trying to bring them out of anesthesia, which we have to do each year to clip the hooves and do the veterinary tests. But all the features of Prader-Willi could be studied in a pig model. Unfortunately, due to cost, 1 day of cost for these pigs was the same as having a hotel room in Pittsburgh. So, it was difficult. And I hope that maybe industry will get behind once, I'm just finishing off the manuscript now. We have frozen fibroblasts, so we can recover the pigs by somatic cell nuclear transfer. And we actually have done that for another project. We generated a pig model, so we know it works. So generating the pig model was efficient, produced many of the neonatal features of Prader-Willi, and with anesthesia complications suggestive of hyperphagia and with scoliosis. So many of the features of Prader-Willi are shown. The difficulties of the pigs, I mentioned the cost. They are still very large even with the mini pig, so it's not easy to work with. There's a long gestation time. It's basically long generation time. It's basically several months of gestation and then 7 months to maturity, so it's basically a year per generation. And so, let's see. Whoops. Okay. So I'm considering other mammalian models, in particular, potentially the cat, because cats also are gyrencephalic like pigs. One advantage of the cat compared to the other models I've talked about is that the Prader-Willi gene cluster is exactly the same as human. There's no difference in gene content. So we actually generated a somatic, just a cell culture model. We deleted the imprinted control region. The Prader-Willi genes are all highly expressed in this cell line and in hypothalamus and pancreas. And so these lines are missing the Prader-Willi gene expression, the SNRF and the SNO RNAs. They retain expression of nectin and Macorin-3 and MAGEL2 because this is a somatic cell model, whereas in vivo we would lose the entire expression. And so that's just something we're trying to see. I'm trying to work on seeing whether we can get funded and work with generating a new model for Prader-Willi. The final part of the talk is we've taken the approach. We tried initially to reactivate the silent maternal allele using epigenome editing, and we could increase expression of the paternal allele, for example, in the pig somatic cell hybrids, but we couldn't, with the approach we took, initially reactivate the maternal allele. And then others kind of have moved much further with that, so we'll let them do that. And so I took a different approach, which was, could we use synthetic biology and miniaturization of the components to try an AAV or adeno-associated virus approach? Problem with AAVs, you can only fit 4.5 kb of DNA in, and the Prader-Willi region is 2 megabases in size. So how could that possibly be possible? And the reason is because many of the Prader-Willi genes are small. So nectin's small, SNRF and SNRFN are both small. These are protein coding. And then all the SNO RNAs are very small. And so I designed a synthetic set of, and we're trying alternative constructs where we have synthetic introns that express the SNO RNAs and these 3 proteins, so we can actually fit within a single AAV 8 of the Prader-Willi genes. We can't fit Macorin-3, but that's a gene that's involved in premature puberty, probably has a very minor role in Prader-Willi, and we can't fit MAGEL2, which is a huge gene and needs its own AAV if you take that approach. But what we're trying to do is use, in our beta cell model, I didn't say it earlier, but nectin and MAGEL2 are not expressed in the beta cell model at all, even in the controls. So we know they don't play a role in the insulin secretion defect and the ER chaperone defect. And so we're using these AAV gene therapy vectors to overcome the cellular defects in the beta cell model and then to inject into Prader-Willi mice to see if we can improve the survival and correct the hyperglycemia and the insulin secretion defects. And another way we're able to do this, the promoter that drives expression of transgenes is quite large, that people use different promoters. I developed a set of miniaturized promoters derived by multiple copies of binding sites for a transcription factor called NRF1, which is actually the major transcription factor that regulates Prader-Willi genes in vivo. The only one it doesn't is MAGEL2. It regulates every other Prader-Willi gene. And so we generated constructs that have from 1 to 10 copies of these NRF1 binding sites. We hooked it up to luciferase, And what you see is that as you increase the number of NRF1 sites, you increase the amount of luciferase production dramatically. And so we focused on the 4x and the 10x copies to do further work. And this is just showing plasmid transfection of beta cells, myoblasts, fibroblasts, schwannoma is a nerve cell. And you see that 4x NRF1 is just as good as the CMV standard large viral promoter, and then the 4x and 10x from the beta cells. So these promoters are driving good expression. And so then with Tina Zhang and my colleague George Giddy's lab, she made AAV vectors in which she linked up the 4x NRF1 and 10x NRF1 to both luciferase and GFP and made AAV vectors and showed that they transduce beta cells. This is the GFP expression and luciferase expression, comparing CMV to AAV8 here, or AAV6, two different serotypes. The 6 is the best for pancreatic beta cells. She also showed high expression in mouse and human alpha cells, like glucagon-producing cells. And then this experiment, what Tina did was inject the AAV vector on the left here into the tail vein, which then goes through the liver, and the liver takes up all the AAV, and so the luciferase expression, this is a whole body luciferase assay, and so it's in the liver, but we wanted to get it to the pancreas. Well, they can inject directly into the pancreatic duct, and then it goes in, and then by dissecting tissues, it's all in the pancreas. So again, in vivo, these promoters drive high expression in the target tissue, which is the pancreas for these studies. And so we're now working on actually incorporating the Prader-Willi genes in these miniaturized vectors to see whether we can rescue those beta cell phenotypes in the animal models. So close, based on pig, mouse, cellular models, Prader-Willi and printed genes regulate metabolic processes. They regulate hormones that are important for growth, eating, metabolism, and reproduction. So these are key functions of Prader-Willi genes. Talk just a little about human genetics. We're going to hear more from David Gogler. He's done some incredible work to establish newborn screening for Prader-Willi and other chromosome 15 disorders. I've been wanting to see that happen for 20 or 30 years, so congratulations with that. You have cell models. We still have cell types to study, such as oxytocin neurons, either to continue to develop the pig model for behavioral studies or other smaller mammalian models like the cat. And then combination of the approach we're taking for gene-based therapies and that others are taking to reactivate the silent alleles, hopefully will lead to new therapies for Prader-Willi. And to thank, particularly the last decade in my lab, Dr. Eric Copies and Marie Johnson have been critical, my cytogenetics collaborator, Suzanne Golan, and her colleague, all my colleagues in Missouri, particularly Dr. Randy Prader, and his postdoc, Bethany Riddell, were critical for the pig model studies. We've had funding from FPWR in the US, particularly also from PWSA, from the Storr Family Foundation. John Storr, many of you may have seen his book on Prader-Willi, and if you haven't, it's an incredible book discussing the sort of history of development of PWSA in the US. And then I've also had much of my NIH funding from NICHD. Thank you.
Fantastic talk, Rob. Thank you very much. And we're off the mark with questions right away. I'm going to pass this mic over and then I'm going to get the other one.
Thank you. That was very interesting. You showed us that your Prader-Willi mice die from hypoglycemia. Now, hypoglycemia leads to low insulin, but you showed us that low insulin was primary In other words, there's a defect in insulin secretion. When you get that, you get diabetes. You get very high sugars, not low sugars. So my question is, has anyone considered what the mechanism of hypoglycemia is in your mouse? And I can tell you that the most likely is that the mouse is unable to produce glucose from the liver.
So so the short answer is no, we haven't got the molecular mechanism. What's unusual about our mice is that glucagon levels are also low, because when insulin's low, glucagon should be high. And so we're just starting now to try to study the glucagon secretion mechanism. It's much harder to study glucagon secretion in alpha cells. There's just many fewer of them. I think you're probably right that we're having a failure in initiating of gluconeogenesis. What I didn't have time to show was that we looked at hepatic glycogen stores, and the Prader-Willi mice store glycogen normally during fetal development. So postnatal day 1 at birth, they have normal liver glycogen levels, and in the rodent, in the first 24 hours, they burn their liver glycogen stores until their nutrition from the mother kicks in. And so the Prader-Willi mice, the reason they have normal blood glucose in day 1 is because they're burning the liver glycogen. They overshoot. They actually drop even lower because they're not picking up, and then they're not recovering, whereas the wild-type mice start then improving the hepatic glycogen. So I think you're probably right that they're not initiating gluconeogenesis in the liver, but we've not been able to look yet further at that.
There's a lot of genes involved in producing glucose de novo in gluconeogenesis. So Pepsi-K is the rate-limiting enzyme. If I still had my lab, I would have offered to measure hepatic glucose output in these mice, as we've done previously. But
that would have been fantastic.
I can't.
We did look at liver in terms of RNA expression, and we didn't see abnormalities in gluconeogenic enzymes. And then at the time, we just didn't have the resources to take that further. And so, sorry. Yeah, I'm not sure about that. I don't know. We have some endocrinologists in the field that could address that.
No, it wouldn't be.
Can we get a microphone to David for the next question?
You're next. Thank you for the great talk, Rob. I've got a bit of a crazy suggestion / question that came upon me just now. So 2 questions that I was considering: how to save your mice, and 2, what is the minimum amount of activity you would require to have therapeutic effect of all the genes? I was just wondering, have you considered a transfusion of wild-type beta cells through liver into your mice in different doses to see whether, 1, your mice will survive with wild-type, and 2, how much you would require for them to survive of all the genes?
Well, that's actually an interesting suggestion, because my colleagues at Children's Hospital Pittsburgh, George Giddy's lab, his lab is they're focused on diabetes, and that's something they're expert at is transplantation of pancreas and islets. So we could consider that. We haven't as yet, and we're hoping to use the AAV gene therapy as one approach, but that would be a really interesting consideration to see if that was sufficient. That's a great idea. I'll have to talk to them, see if they're interested in trying that.
Hello. Here. Very nice talk. Have you checked the level of glucagon-like peptide 1? Because this one is when in response to the meal, the intestine will produce the GLP-1, and this will control the insulin secretions, and this is related to the hyper. Yeah,
that would be something that's good to do. We haven't. Our our deletion, it's a long story, I won't go through it, but we about a decade ago, we lost our deletion line of mice. And I've just imported, a couple months ago, the imprinting defect mice from Jim Resnick in Florida. And so we're starting to work with them now, but we're still establishing the strain in my lab. And then we're using that for the gene therapy approach. But I'm also hoping to be able to greatly extend the physiological analyses. So GLP-1, glucagon mechanisms, other liver, hepatic, those are things we hope to be able to study then with that mouse model. Because most people are focused on the brain and nobody's really looked at metabolic tissues like pancreas and liver.
Hi, I have a quick question. Thank you so much for a fascinating talk. I'm fascinated by this potential, the mechanism that you've discovered potentially explaining not just one, but the plethora of Prader-Willi syndrome endocrine defects that remain incompletely explained. And those low endoplasmic reticulin chaperone proteins that you found, like the HSPA5, have you been able to link those back by any chance to the molecular mechanisms in Prader-Willi syndrome, like maybe targets of SNO RNAs or something that links the mechanism?
Have we been able to do what, sorry?
So link those low levels back to the molecular mechanism of Prader-Willi syndrome, maybe targets of the SNO RNAs or something that links?
Other than the pancreatic beta cells, no, we've not looked at that, and we haven't gone back to human. I've been wanting to get pancreas from human to study, and the trouble is that in the US, the brain banks have brains from Prader-Willi subjects. Nobody collects pancreas, and it's also extremely difficult because it's full of digestive enzymes. So if it's not done right and quickly, there's nothing left to really look at anyway. And so I also wanted to recreate the Prader-Willi genetic defect in human islets, and I've been trying to seek funding for that, but I haven't yet been successful.
That would be fascinating.
And so that would be the alternative, since we don't have the islets or the pancreas from patients, was to recreate it. We have the technology to do it. I just need the funds.
I look forward to reading your paper, and I look forward to finding out, maybe your group will find out over time whether this whole folding and secretion defect is more widespread. That would be fascinating to find out.
Exactly.
Thank you for a wonderful talk. Now, I just have a question about the pig models. We've heard a lot about apraxia, speech apraxia and gait apraxia in Prader-Willi, and I was just wondering if you're seeing any motor signs or motor coordination issues in your animal models.
We did not, but we didn't yet look for that. The problem is that, as I say, the studies were expensive. We had an N of 1, so we really didn't have a control set of pigs and enough animals to do carefully designed studies to look at. We wanted to look at hyperphagia and all these other aspects that can be studied. And so right now I don't have live pigs. We have cryopreserved newborn fibroblasts, and so we can use somatic cell nuclear transfer to regenerate. What's really interesting is, somatic cell nuclear transfer, or cloning, generates identical organisms to the one you're cloning. In this case, if we clone our mosaic, we actually will get two completely different types of pigs. We'll get the Prader-Willi pigs, and we'll get the wild-type controls from the same litters. And so it's actually perfect. And for another project, we actually generated a pig model of phenylketonuria, PKU. For that project, after we published, a company got behind and funded a lot of research, and we've generated dozens of clones of the founder PKU pig. So I'm sure it should work for Prader-Willi. I just need funds. Hopefully industry or something will get behind. The problem is that whenever I've talked to industry with Prader-Willi, they're always like, well, what's the mechanism? What's the cause? And when you say how complex it is and you don't know the ultimate cause, then they're like, well, that's not clear how we're going to make money from developing a hyperphagia or obesity drug or something. And so it's been very difficult to try to get the support. I'm sure that's familiar to everyone.
Thank you.
Excuse my ignorance around deletions and that. Is there a difference? Do some people have bigger deletions or smaller deletions? And how does that affect them?
Yeah, there's largely two sizes of deletions because of these common breakpoint hotspots.
Yeah.
So there's a larger deletion that's like smaller. There's 4 functional genes that differentiate those, all of which are expressed in the brain. And so there's been some types of phenotypes assigned to those, but those are recessive genes. And so missing one copy shouldn't make that much difference. So it's not clear yet still, despite multiple studies, whether or not there's a difference slightly small. But by and large, all the same imprinted genes in both groups.
And also, like Angus was tested 25 years ago and he has a deletion. Would he, with the advancement in testing, would he need to be retested, or he's a deletion, he won't be anything else?
That was from chromosome studies?
Yes. The fish.
Yep.
And there was a second one, and he was positive to, both.
Yeah, so there's probably no need because the deletions, because of this mechanism, they're essentially all the same size. And yeah, congratulations on everything you've achieved. It's really incredible. So,
I'm curious about the mosaic, Peg, and whether that can teach us anything about how much gene expression we're going to need to achieve if we successfully reactivate genes.
That's a really interesting question. And we tested many tissue types, and it's essentially 50% of each cell type in lots of different tissues we tested. We haven't done fine analyses of sub-brain regions, but in all other tissues, it's 50%. So we're quite certain that the mutations occurred at the 2-cell stage, so that we generated the 2-cell lines. And he does have a much milder phenotype. And so the one aspect, so either that means that 50% increase, you don't have to go the full way, that just some increase in some subset of cells will be sufficient, with the one proviso that we haven't specifically studied, hypothalamus or something else, and so whether or not it's possible that in a tissue like that the number of cells with the Prader-Willi defect are much less. And so maybe in that region there's 80% of wild-type cells. We don't know that.
Fascinating. Lots to learn. Last question.
I'm not a scientist. Rob, the increase in UPD from 25 to 40%, when I look at that, is it correct to interpret that as an increase in incidence rates then of Prader-Willi overall? And is it a 15% increase?
Yeah, I don't think it's an increase necessarily in Prader-Willi overall in the population, but what it is in certain populations where parents are having children at a later age, particularly mother in this case, that we know that the mechanism is via an initial trisomy 15 and then trisomy rescue, because trisomy 15 is not compatible, at least if it's not high-level mosaicism, with survival of the fetus or embryo. And so it's the same mechanism as Down syndrome. So in some parts, there's been one study, I think it was from Switzerland, that showed up to over 40%, maybe up to 50% of the Prader-Willi cases had UPD. And that's because the parents, the woman, were older at the time. So I don't think it's affecting the overall population incidence. But then again, it's, when David's work can be, with newborn screening, can be taken worldwide, and we can really look at true population incidence, then we can actually see whether there's any association.
So I just think about the incidence of deletion, and is there a reason to think that the incidence of deletion is going down in the total population? Anyway.
Well, the deletion, occurs randomly, but there is good reason to suspect that there are potential environmental effects. So if you get an increase in the deletion, occurs because of misalignment and combination of DNA segments, and that if you increase the frequency of double-strand breaks, which some chemicals do, then that can increase the frequency of the deletion event. So there have been some past studies some time ago that suggested a potential association of the Prader-Willi deletion and hydrocarbons. There's a lot of other work not related to Prader-Willi, but that shows increases in recombination between duplicated segments due to things like hydrocarbons and other chemicals. So certain types of environmental exposure could lead to increases